Molecular Biology by Joanne Nazario. We're going to begin talking about DNA and some basic properties. DNA is known to be double-stranded, which means there are two strands rather than one. The building blocks of DNA are nucleotides. They are known to be the protein manual, and they must be placed in correct order. Nucleotides are used to read and further transcribe and translate proteins. These are represented by four-letter alphabet: A, T, G, and C. These stand for adenine, thymine, guanine, and cytosine, which are all considered the bases. Within these nucleotides are ribose sugars and phosphates. That is known to be the backbone of these nucleotides. They also repeat as the bases change. The bases change, but the ribose and phosphate groups remain the same. Nucleotides all contain 5' and 3' ends, which allow for the phosphate group is found off the 5' carbon of the ribose, where the hydroxyl group is coming off the 3' end of the ribose. This phosphate group gives the backbone a negative charge. The 5' carbon will always be bound to the 3' carbon of the next nucleotide. Adenine will always base pair with thymine. Between adenine and thymine, there are 2 hydrogen bonds. Cytosine will always base pair with guanine, and between those bases are 3 hydrogen bonds. This image allows you to see the representation of the base pairing along with the phosphate and ribose backbones. You can also see how DNA is double-stranded and how it creates this double helix. The next thing we will talk about is chromatin compaction from protein-free DNA to mitotic chromosomes. Chromosomes are long DNA molecules that are condensed together. In order to transfer and use these chromosomes, they must be accessible in small compartments rather than loose strands that may intertwine. With relaxed strands, there is accessibility to create copies of the genes of the required sequence. But with compaction, it allows for all chromosomes to have their own zone or region and avoid being tangled. The manner in which we reach this compaction is done by several mechanisms. The one we're discussing now is the use of nucleosomes. Nucleosomes use histone proteins to create chromatin complexes. Using histone H2A, H2B, H3, and H4, there's a creation of an octamer when you make copies of each of those proteins. This complex uses 8 proteins and creates a spool shape for DNA to bind around. This shape is influential for condensing DNA. Long strands of DNA will wrap around the octamers in a continuous fashion and will receive compaction. To define nucleosomes, all these compartments must be present: histones, octamer, wrap DNA, and linker DNA. The DNA strand that is revealed and not intertwined is known as the linker DNA. We know that DNA is negative, and it binds to the histones due to the histones being largely positive. This positive comes from the lysine tails found on the histones themselves. If we look at this visualization of histones, we can see the histone octamer created here by all 8 histones. You can also see how DNA is twirled around the octamer. As well, you can see the linker DNA between each histone octamer. You can see all the defining components of a nucleosome. You can see the octamer, the DNA bound to the octamer, and the linker DNA in between each. Here, you can see how the compaction is done. When you see here, you can see the octamer and the DNA bound around the octamer. You can see the linker DNA again, and the next connecting octamer. Each bead is going to be turned and swirled in a fashion here, using this H1, this histone 1 protein. When this histone 1 protein is present, it allows for the bead itself to be turned and tinkered. If you see here, you can see how DNA was bound around each octamer, creating this nucleosome beads, and then are then tinkered and turned using that H1 protein. Here you can see the process from DNA to nucleosomes to this 30 nanometer fiber, which contains all those twirled nucleosome beads. From here, you will continue to compact and compact and compact until you have this chromosome like you see here. Now we can discuss chromatin remodeling. We already are aware that DNA is tightly bound due to the charges found on the histones, positive, and DNA being negative. We discuss here the use of hats, HDACs, and this histone complex shift. Hats are histone acetyltransferases. HDACs are histone deacetylation complexes. When lysines are acetylated, it allows for the positive charge to be neutralized and weakens the bonds between the nucleosomes and DNA. When lysines are deacetylated, the positive charge is as strong as it was prior. Histone acetyltransferases are acetylated histone tails, and histone deacetylated complexes are deacetylated histone tails. In order to loosen the chromatins, there are chromatin remodeling complexes. These complexes move nucleosomes out of the way by using the force exerted from ATP hydrolysis. This force allows for the complexes to unwrap DNA regions from some octamers. In this picture, you can see how the unacetylated are tightly bound. When you use hats, now containing acetyl groups, you can see how it's a little looser and you can reach some of the sequences on the DNA. When you add the HDACs, you will be removing those acetyl groups and tightening up that nucleosome structure again. In this image, you can see this ATP used in order to slide the nucleosomes and unwrap and loosen pieces of those of that strand.